organisms that still have, that start to have these tissues. We talk about the cnidarians, like jellyfish. Another lineage are the ctenophora. These are sometimes referred to as cone jellies, sea walnuts. They say they're referred to as sea gooseberries. I've never heard them referred to as that, but maybe someone somewhere calls them that. So if you want to call them that, please feel free to do so, but just make sure you're not talking about this phylum, Ctenophora. Oftentimes these are confused with jellyfishes. They look the same. They look similar as the Medusa stage of the cnidarians, but they are a distinctly separate phylum. That's one thing that a lot of biology students have a hard time realizing initially, is that you can have organisms that look the same, but they're going to be in different phylum, they're going to be in different orders. So just because something looks the same does not mean it's the same lineage. How many people think that spiders are insects? They are not. Insects is an order, or a phylum, class, insects are a class, arachnids are another class. So again, knowing this phylogeny tells you how these organisms are related. Insects and spiders are all arthropods, but they're in different classes. One of the ways that you can recognize the ctenophora from the cnidarians is by looking at the cilia that they have. Cnidarians don't really have any cilia. The ctenophora have 8 rows of plates of cilia that beat together that enable them to move. How do the cnidarians move? What do they do? They have rhythmic contraction based on muscles. Here, the ctenophoras have the cilia that move them around. So the cone jellies don't contract or the body of the organism themselves. Many of these are bioluminescent. You can sort of see this one glowing here. Bioluminescence is something that has evolved multiple times in different lineages. They do have some tentacles. These tentacles are covered with cells that are called coloblasts. These are used for capturing prey items. However, unlike cnidarians, where they have the nematocysts that release sort of that toxin, that venom that can paralyze the prey, these basically just have a glue. There's an adhesive that these tentacles release, and it physically holds on to the prey. It doesn't kill it, it doesn't paralyze it, but it makes it impossible for that prey item to escape. The ctenophore then starts the digestive process. Now, the phylogenetic position is somewhat unclear. They are their own phylum. They used to be grouped very closely with the cnidarians for obvious reasons that they look very similar. But genetic evidence has sort of thrown that up in array a little bit. They're sort of uncertain what organisms these might be closely related to. So that sort of brings us to the end of the organisms that are not bilaterally symmetric. We started off with the non-symmetric organisms, the Porifera. We talked about the radially symmetric organisms. Now we're moving on to the bilaterally symmetric organisms. Remember, we're sort of going through in terms of these evolutionary advances. These are all characterized by having bilateral symmetry. As we talked about, bilateral symmetry allows for high levels of specialization. You can have directional movement. You can have concepts of moving forwards or backwards. You have cephalization, the development of a head-like region. It allows for more articulated control of limbs. So overall, bilaterally symmetrical organisms have more specialization, more efficient processes. Now, bilaterally symmetric organisms are generally classified by the condition of their coelom, meaning that we look at what's going on with the body cavity. Some of these are called acoelomates. Acoelomates don't have a coelom. So you have bilaterally symmetric acoelomates. You have bilaterally symmetric pseudocoelomates, organisms that have the fake body cavity. It's not a true coelom because it's not surrounded by mesoderm tissue. Instead, it's a separation between the endoderm and the mesoderm. And then you have the bilaterally symmetric coelomates. So right now, we're going to be focusing on the acoelomates, the bilaterally symmetric organisms that still are lacking a true body cavity. The first of these are the phylum Platyhelminthes. These are soft-bodied organisms. They're ciliated. They oftentimes have cilia that enable them to move. But the bodies are completely solid inside. There is no internal body cavity. If you were to dissect one of these, you would see tissue throughout the entire organism. Many of the Platyhelminthes are parasitic in nature, meaning they're going to be parasitized use energy resources, get food resources from some other organism. There are some, however, that are free-living that are primarily going to be found in soil environments. Some of them are also found in marine environments, freshwater environments. The key thing you need to know is that they need to generally have a moist environment. If they're found in soil, it needs to be moist soil. They're not going to be found in the middle of the desert or something. So they're moving around in any sort of moist environment. Now, one of the key interesting features about the platyhelminthes is they have only one opening for the digestive cavity. This is the same thing we saw with cnidarians. There's one opening where food enters, and then the waste products also leave. They do have muscular contractions around a pharynx region, so we have a little bit more of a developed digestive cavity. It's not just a hole that is found inside the organism. So the food gets moved around in the digestive cavity. It allows the food to be ingested. One thing when we talk about the digestive system, the muscles that are found there are also helpful in breaking up the food. We have something like teeth. Other organisms do not have teeth. They can't break the food up, so they rely on the muscular contractions to sort of tear the food into smaller pieces. Even though they have a fairly well-developed digestive system, they do not have a circulatory system. So what this means is anything that's moving through their system has to move through the process of diffusion, including the gases that they need to survive. They're going to absorb oxygen directly, it diffuses through the organism, they release carbon dioxide. So the gut, the midgut, is responsible for helping diffuse the food through. They do have a gastrovascular cavity. Food is moving in, but once it's digested, it's going to diffuse through the gut and diffuse through the organism. The particles of food are digested extracellularly. That's what we saw with the cnidarians. We've continued that trend. It's an evolutionary adaptation that the vast majority of animals employ. So the cells engulf the food particles through the process of phagocytosis, or engulf the nutrients through phagocytosis. Some examples of these would be, the one difference that is separated from this are tapeworms. Tapeworms are another form of platyhelminthes, but they completely lack a digestive system. Where are tapeworms found? In the intestine, in another animal's digestive system. So they don't need to have a digestive system. The animal where they're found is breaking the food down for them. All they need to do is absorb the food, absorb the nutrients directly through their cell walls. In addition to a digestive system, they also have an excretory system and an osmoregulatory system. So we start to see the evolution of organ systems, tubes, and structures running through the entire body of the animal. Now, metabolic wastes are being produced. They are secreted into the gastrovascular cavity, but because they only have one opening, they don't have an anus. It's not a tube system like we have with some other animals. The wastes are excreted through the mouth. They also have a simple nervous system. So we're starting to see the evolution of a more complex nervous system. There's a region in the anterior portion of the animal that has sort of a congregation of nerve cells referred to as the cerebral ganglion. There's a nerve cord that runs through the organism. So it's not just a collection of nerve cells. We start to see a specialization. Again, anytime we have specialization, it's going to allow for more advanced features. One thing we see with these is they actually have eye spots. Not true eyes like we think of with vertebrate eyes or insect eyes, but they have an eye spot that is capable of distinguishing light and dark. And this is a fun experiment that sometimes is done in the lab. You have like a petri dish where it's half light, half dark, and you can see the platyhelminthes moving to different areas. They have reproductive systems. Most of the platyhelminthes are hermaphroditic, meaning they have both both male and female sexual reproductive organs. So they do undergo sexual reproduction. The organism is going to be producing male gametes and female gametes. The difference is you simply have one organism that is producing both of them. They are also capable of undergoing asexual reproduction. Things like the flatworms are capable of budding and regenerating body parts. So if you split one of them into two, they will regenerate. Now, as we said, they're also solid mass of tissue. If you cut one in half, you're not going to see any sort of body cavity. The only opening that you have is this gastrovascular cavity where food is ingested and digested. You do start to see, as we said, the different body structures. We see reproductive organs. We see nerve cords. This is where the pharynx is, where food is ingested. So we start to see a more specialized body system. If you look through it sort of from the top, you see that they have an excretory system. Their intestines running around that are going to help diffuse food products, not gas, but food products through the organism. Also pick up waste. They have a nervous system. We see a congregation of the nerve cells of the anterior cerebral ganglion, and then the nerve cells run throughout the entire organism. And then we have a developed reproductive system, both male and female reproductive organs. Now, the platyhelminthes are broken down into two major lineages. Some of these are free living, meaning they're going to be found in any kind of environment. These are referred to as the tubularians. These include the planarians, the stereotypical platyhelminthes that we see with the two eye spots right on top. Planarians are oftentimes used in laboratory experiments. They're very easy to rear. They engage in some very simple behavioral differences. But you have a second lineage of platyhelminthes that are parasitic. These are called the Neodermata. These are primarily called the trematodes and the flukes. So these are parasitic organisms that are living inside other organisms. They're internal parasites. You have some that are external parasites that are attached by hookers, but many of them are going to be found on the interior of the organism. And one thing that's interesting about these organisms is that they have multiple life cycle hosts. They have to undergo certain developmental processes in one host, then they have to move to another host to undergo certain other developmental processes. So it's not that they're simply parasites of one organism. They're going to parasite, in some cases, 2 or 3 or even 4 organisms in order to undergo their complete life cycle. Now, an example of one of these are the tapeworms that we mentioned, the Cercomeromorpha. These are the ones that are found in the intestines of animals. But a lot of the flukes have life cycles like this, where they might start off in one organism, say a snail. They undergo certain developmental processes. Eventually, that mollusk might be eaten by a fish. They then undergo certain additional processes in here. That fish then might get eaten by some vertebrates, such as ourselves, and they'll finish their developmental process. So it's a 3-host life cycle. They're going to be found in 3 different hosts. If they don't move from one host to the other, they're not going to be able to continue their development. They're not going to be able to continue growing. So this is an obligate 3-host parasite. There's a lot of research that's also being done on these trematodes and flukes because they also engage in something that's referred to as behavioral modification. If you are a trematode or a fluke that's in a snail, you really need to make sure that that snail is going to get eaten by a fish or by a bird or whatever the next host may be. And what these trematodes will do is, through an unknown mechanism, they will alter the behavior of their host to encourage it to be passed along to the next generation. The classic example of this is when you have a trematode that is infecting a fish that needs to move to a bird as the next host. What do you think that trematode might induce the fish to do? Jump out of the water. That's a good idea. Similar idea, they actually end up swimming near the surface. They slow the fish's movement down. And now fish have a very easy way of getting away from birds. Go down under. Bird flies by, fish goes under, not going to be able to get eaten usually. But if there's a fish that's moving slowly, swimming near the surface of the water, that's a prime target for birds. And what they've shown experimentally is that fish that are infected with certain kinds of trematodes are more likely to swim slowly, are more likely to swim near the surface of the water, and that certainly is not good for the fish, but it is very good for the trematode. There are other examples of behavioral modification. This is really a hot area of research because here you have this very simple organism somehow controlling the behavior of a much more complex organism. Now, the Cercomeromorpha is another one that I mentioned, the tapeworms. These are ones that are purely found internally. They hang onto the inner lining of intestines. As I mentioned, they don't have digestive tracts because they have nutrients all around them. They just absorb it directly. The next phylum after the platyhelminthes are the Acoelomorpha. Acoelomorpha are similar in some fashions. They have a very primitive nervous system, but they differ from platyhelminthes in that they lack a digestive cavity. So again, we start to see simple evolutionary changes, but there are differences between the different phylums. They were previously thought to be related to flatworms. Again, morphologically, they're very similar, but molecular evidence suggests that any similarities that we see are examples of convergent evolution. The organisms live in similar environments, they're similar sizes, similar environmental pressures, and so similarities are due because of this convergent evolution, not because of common ancestry. Recently, a third phylum was discovered, phylum Cycliophora. This was discovered in 1995. Prior to this, it had never been found before. Now you might say, well, why would no one ever know about this? It's because of where these live. These organisms live on the mouthparts of lobsters in the North Atlantic Ocean. So you can sort of see why it might have been overlooked a little bit. Not too many people are sitting there looking at it with microscopes at the mouthparts of lobsters. But they did find some of these in 1995. They reported them. We now have a new phylum of bilaterally symmetric acoelomates. They're very different from all others that we know. There's one thing that's interesting is they have this ring of cilia around the mouth. It's thought to aid in the feeding process, sort of filtering food out. But basically, they're relatively sessile. They don't really move around that much. They basically are attached to the mouth structures of the lobster. So those are the acoelomates. You then have two different phyla of pseudocoelomates, bilaterally symmetric organisms that have a pseudocoelom. So we start to see a body cavity, but it's not what we consider a true body cavity. We still have the three tissue lines, the endoderm, the mesoderm, and the ectoderm. But with pseudocoelomates, the body cavity lies between the tissues that are the mesoderm and the endoderm. So we have a separation of the two tissue types, creating a body cavity. Otherwise, that body cavity is very similar to what we would consider a true body cavity. It's filled with a fluid, like we see in a true body cavity. The fluid performs a lot of the functions that the circulatory system does. In organisms that have a true body cavity, most of those have a circulatory system, but the pseudocoelomates have this body fluid that sort of circulates through, helping move materials and diffuse materials through. It is not a monophyletic lineage. If you look at this phylogeny, the two lineages are the Rotifera and the Nematoda. If you go back to the most common recent ancestor, there are other things that are not pseudocoelomates. So it's thought that pseudocoelomates, the evolution of this pseudocoelom, might have arose independently in both of these lineages. If this was due to common ancestry, you'd expect to see more pseudocoelomates in those lineages. Because you just see two separate lineages, it's probably evolved independently. So again, quick overview, you have the platyhelminthes things, these are the acoelomates, no body cavities. You have the pseudocoelomates where you have a separation of the endoderm and mesoderm. So let's talk about the two phyla. One of these are the nematodes, often referred to as vinegar eels, eelworms, or roundworms. Roundworms, although is technically incorrect, because there are organisms that are specifically roundworms. These are very diverse in terms of where they're found. They're found in almost any kind of environment, marine environment, terrestrial environment. They're going to be parasites, they're going to be free living. So they're going to be very common in almost every kind of habitat. These are going to be bilaterally symmetric, they're not segmented, and they're going to be pseudocoelomates. They are covered with sort of a thick, flexible cuticle. They do actually have to molt this cuticle as they grow and develop. Anytime you have an organism that's covered with a relatively strong barrier or a strong protective covering, if that organism is growing, more often than not, they need to molt that protective covering. The covering itself is not going to grow and expand. So in some ways, they share similar functional features with things like arthropods, but they are pseudocoelomates, while arthropods are true coelomates. They have a very well-developed digestive tract. The evolution of the coelom is a more advanced evolutionary characteristic, so we're going to see more evolutionary advanced traits within these lineages. They actually have a stylus that is a piercing organ that's near the mouth. This is used to sort of stab or pierce prey items and make sure the prey is not able to escape. They have a pharynx that then creates a sucking motion so they can suck the prey food item in. And then they do have an anus. So we now start to see the evolution of a true tube-in-a-tube system. You don't just have one opening for the gastrovascular cavity. Food is going to enter in one end, waste is going to exit out of the other. So if you sort of look at it from the side or cross-section, you see this pseudocoelom here. There is this body cavity. You have the endoderm in the middle. You have the mesoderm surrounding it. Between those is where the body cavity is found. You see that they have fairly well-developed organ systems, including this digestive tract. You have the mouth, and then on the other end you have the anus. They do engage in sexual reproduction. You have male organisms and you have female organisms. And they're actually sexually dimorphic. You can look at these and identify whether something is a male or a female. Males have are generally smaller, and they actually have a small hook that's at the end of the tail. It's thought that this hook aids in the process of reproduction. Fertilization takes place internally. So the males release the sperm and fertilize the females internally. Now, there's a lot of different behavioral, ecological niches that these fulfill. Some of these are active hunters. They primarily prey on produce, other very small animals. Some of these are parasites of plants. So others are parasites of animals. So there are going to be a lot of different kinds of adaptations to the specific behavior. Some of these are causing diseases within humans. There are about 50 different nematode species that cause disease in humans. These include hookworms. They include trichinella, that causes trichinosis. Elephantiasis is caused by nematodes. Filariasis is caused by nematodes, things like African river blindness. Pinworms, intestinal roundworms, these are all examples of nematode disease. The other phylum, aside of Nematoda, are the phylum Rotiferans. These are again, bilaterally symmetric, they're unsegmented, they have a pseudocoelom. They also have very highly developed internal organs. They're recognized by a ring structure they have called the corona. It looks sort of like a wheel that's found on the organism, so they're referred to sometimes as wheeled animals. The ring structure is made of cilia that's found on the anterior end of the animal. It's used primarily for locomotion, to help move the animal around, but it can also help sweep, move, or create a current to move food to the mouth. So this is what it looks like. You see the cilia here, the sort of the wheel structure right here. This can help, the cilia can move and help swim, the organism swim around, but it can also sweep food towards the mouth, and where it's ingested, and again we have this tube-in-tube system. You see the actual evolution of a brain structure now, not just increased cerebral ganglia. You see different organ systems within the rotiferans. So that brings us to the end of what's going to be covered on Test 1, which will be open up on Monday, March 18th. It's going to cover 5 chapters, 36, 38, 42, 32, and 33. So 36 was plant structure, 38 was the plant movement of material through plants, 42 was plant reproduction, 32 and 33 are starting sort of the overview of animal diversity. Now, it's going to seem like very quickly after we have Test 2, we're going to have Test 3. But the reason for this is because Test 2 is now going to be delayed until after spring break, and we're going to still be starting the material for Test 3. So it's going to seem like there's a short delay between those, or a very short delay between those two. Now, Chapter 34 was included on the Learn Smart module. I have extended that due date, so Chapter 34, Learn Smart homework assignment, will be due on the same day that we have test number 3. Any questions about this? All right, so with Chapter 34 now, we're going to be talking about new different organisms. We're leaving behind the acoelomates, the pseudocoelomates, the radially symmetric organisms. Everything we talk about from now on is going to be a true coelomated organism. They're all going to be bilaterally symmetric. The way that we split the next lineage of organisms is into whether they're invertebrates, meaning they don't have a vertebra, or whether they are vertebrates. So the invertebrates lack a vertebra. There are lots of different lineages of these. Anything that is an animal that we haven't talked about that doesn't have a backbone is going to be found in the invertebrate lineage. They're all going to be coelomates. They're all going to be bilaterally symmetric. They're all going to have the 3 tissue types. We're going to start to see the evolution of more advanced structures within the animals as well. more organ systems, more evolution of different kinds of, or specification of tissue types. So these are overall going to start to be more what we think of as actual animals as opposed to some of these other animals, which if you talk to a non-biologist, they're not going to be sitting here contemplating. So we have a quick video here that we'll watch, then we'll continue on.
Crabs, worms, cuttlefish, corals, insects. These creatures may not look alike or act alike, but they do share one thing in common. All of them are invertebrates, animals without backbones. Invertebrates inhabit every ecosystem, roaming every continent and swimming in every ocean. They comprise about 95% of all living animals on Earth, outnumbering vertebrates almost 30 to 1. Ranging in size from tiny mites to giant squid, the diversity of this group is impressive. Having no backbone is something they have in common, but just because they're spineless doesn't mean they lack support. Many invertebrates like this scorpion rely on a hard external skeleton. Like a coat of armor, it frames and protects the body. Others depend on the pressure from fluids. Water fills the internal canals of this comb jelly. It keeps its shape at depths that would crush us. The sea anemone supports itself by circulating water through its soft body. Worms are held together by a tubular wall of muscle that surrounds their fluid-filled segments. The wall also helps them move. Invertebrates have some remarkable survival mechanisms. Here in the desert, freshwater fairy shrimp hatched from eggs that were dormant as long as 25 years. The size of dust specks, the dormant eggs hatch soon after a heavy rainfall. At another extreme, ice worms make special proteins that prevent deadly ice crystals from forming inside them. It's an odd place to live, but they manage just fine. A few invertebrates can even regenerate vital parts. During a battle, this octopus loses an arm to a moray. But in a couple of months, another one will grow back. We may not realize it, but vertebrates like us owe a lot to the invertebrate world. Insects pollinate plants. Spiders kill unwanted pests. Worms add nutrients and air to the soil. And crustaceans like shrimp and crabs are tasty to eat. The list goes on, and so do the wonders of these incredible animals.
Now, one thing I want to specify here is when we talk about invertebrates, what do they show up there? Well, just some examples, not characteristics.
Scorpion.
Octopus, scorpion, jellyfish. Wait a minute, jellyfish. Are jellyfish invertebrates? Yes, they are. Are they coelomated invertebrates? No. So when we talk about invertebrates, the classification of an invertebrate is a broad classification, basically meaning anything that doesn't have a vertebra. Okay? That includes the acoelomated invertebrates. That includes the pseudocoelomated invertebrates. It includes the radially symmetric invertebrates. So even though those, we've now finished talking about those are still considered invertebrates. If you look at chapter 34, we're now going to be moving on to the coelomated invertebrates. Okay? So make sure that that's clear. You can call those invertebrates, but they're not going to be considered coelomated invertebrates. So one of the most common lineages of coelomated invertebrates is the phylum Mollusca. In terms of animal diversity, this is the second most diverse animal lineage on the planet, second only to the arthropods. This includes things like the snails, the slugs, clams, squids, octopi, and a variety of other organisms. There are going to be certain characteristics that we recognize all mollusks as having. Shells is not one of those. Some mollusks have a shell. Things like snails, certain bivalves have shells. Other mollusks do not. Some mollusks have remnants of a shell. They may have some sort of internal structure that at one point probably was a shell, but it's been lost over time. So the diversity within the mollusks is huge. There's a lot of different types. They live in aquatic environments, freshwater environments. They live in marine environments. They live in terrestrial environments. So they're going to be found just about everywhere. They range in size. You have some mollusks that are basically microscopic, and then you have some that are huge. One of the largest mollusks out there is giant clams that may weigh up to 270 kilograms. Just recently, they've actually developed photographs of giant squid. So the size of mollusks is going to vary. It's thought they evolved in the oceans. This is where we see a lot of groups of mollusks. The vast majority of mollusks have remained in the ocean, but we see many lineages that have spread to freshwater environments, many lineages that have spread to terrestrial environments. They are an important source of human food. As the video said, there's a lot of mollusks that we can eat, certainly certain invertebrates that we can eat, but we can also eat a lot of different kinds of mollusks. They are economically important in other ways. Things like pearls. What girl doesn't like to get pearls? Thank a mollusk for that. Mother of pearl, for people who are maybe a little cheaper. That's actually made from a shell. Thank a mollusk for that. They're also bad mollusks. We have pests. The zebra mussel is an invasive mollusk that actually invades ship ballast tanks. It is microscopic as an immature, then it settles down, starts growing into a mussel shape, what we sort of think of with the bivalve structure. It then starts to weigh down the ships and make the ships unable to exchange their ballast fluid. So there are pest mollusks as well. Now the generalized mollusk body plan, all mollusks have what we refer to as a mantle. They have what we refer to as a foot. The mantle is the epidermal covering of the mollusk. What it may look like may vary from mollusk to mollusk, but there are parts of the mollusk that we classify as the mantle. The mantle sort of bounds or surrounds what we refer to as the mantle cavity. It's an internal cavity that mollusks use for a variety of reasons. The mollusks that have a shell, the shell is secreted by the mantle. So the mantle secretes chemicals that will then harden and become the shell structure. Even if mollusks don't have a shell, they still have the mantle helper. All mollusks also have what we refer to as a foot. The foot is the primary means of locomotion, as you would expect. What does the foot do? It moves you around. But you can have specifics that vary from mollusk to mollusk. So all mollusks have this very muscular foot region. In some mollusks, like a snail, it's a very large, singular, flat region that moves through contraction. In other mollusks, like an octopus, the foot is broken up into tentacles that can move around and help propel the mollusk forward. But that whole region is classified as a foot. All mollusks have internal organs. We start to see the evolution of a fairly advanced internal organ system. And one thing that's interesting is even though mollusks have a coelom, the coelom is reduced. We mentioned that they have a mantle cavity. That is not the same thing as the coelom. The body coelom, the body cavity in mollusks, is a vastly reduced structure. It's primarily found in an area around some of these organ systems, around the excretory system, around the heart, and parts of the intestine. This is unlike something like a vertebrate where we see a body cavity found throughout the whole organism. In addition to those organ systems, they have a digestive system, they have reproductive organs. These are all going to be primarily located in the visceral mass, the solid mass of tissue where the coelom is not found. Now, if you have an aquatic mollusk, they have something that's referred to as ctenidia. These are basically the equivalent of gills. If you live in an aquatic setting, you need to have some way of extracting oxygen from the water. Gills generally work very well, so the mollusks have evolved their own gill structure. In bivalves, things like clams and mussels that have the bivalve shell structure, the ctenidia are also involved in extracting food. They help filter food out of the water. So this is sort of the generalized mollusk structure for a lot of the different mollusks. You can see the foot structure. You can see the gills. A lot of mollusks have a radula, which is near the mouth structure. You have the mantle cavity that's surrounded by the mantle, and then the shell structure. So you have things like the chitons, the gastropods, bivalves, cephalopods. And so these are all going to have all of these fundamental structures of the mollusks. The difference is how those structures have evolved over time to become specialized for each one. So we'll continue with this when we get back from spring break. Everyone enjoy your spring break. Don't forget to study.